Hello and welcome to New York City Atheists live on tape, brought to you by New York City Atheists Incorporated. I'm your host, Dennis Horvitz, the David Letterman of the Hopelessly Damned. Several years, President Obama has lifted many of the bans on stem cell research done by federally supported laboratories, but not only has the United States, and it holds great promise for many diseases and conditions such as diabetes, Alzheimer's, and even heart disease. And it can be involved in rebuilding hearts. But that's not the only, the migration of jobs is not the only problem. Are migrating to these countries. Regenerative medicine basically focuses on stem cell cures, and it holds great promise for many diseases and conditions such as diabetes, Alzheimer's, and even heart disease. And it can be involved in rebuilding hearts. But that's not the only, the migration of jobs is not the only problem. We have what are called, so-called ethical considerations. There are many religious organizations that oppose the use of stem cells, probably in some cases because of very honestly held beliefs, but I'm sure in a lot of cases it's because of their specific religious agenda that they wish to impose on the general public. So one of the ethical considerations that I think we should be considering is the fact that there's a lag in science education in the United States. So here today to address this issue, possibly pose some possible solutions, is Dr. J. Peter Gergen, who is a professor in the Department of Biochemistry and Cell Biology and director of the Center for Developmental Genetics at Stony Brook University. And he has a list of credentials longer than my excuses for not having any. He is a graduate of the Massachusetts Institute of Technology. He has earned his PhD at Brandeis University. He did his postdoctoral work in developmental genetics under a Nobel Prize winner, and he told me how to pronounce his name, and I'm going to blow it. Eric Weishaupt. Is that correct? No. He will correct me when Weishaupt. Eric Weishaupt, who is famous for his work on fruit flies. In any case, before I ruin this introduction any further, please welcome Dr. J. Peter Gergen.
Okay, thanks again for the introduction. As Ken mentioned, my name is Peter Gergen, and I've been at Stony Brook. I just passed my 20th anniversary of being a resident of New York State and on Long Island. I wanted to start off by spending one or two slides just telling you a little bit about who I am and how in the heck I got picked to talk about stem cells. Some personal background. I got my PhD in biochemistry a while ago, last century, and got interested. That was around the time that molecular biology, the ability to clone segments of DNA out and manipulate them in the test tube, was being invented as a field. And it was very clear from the beginning that this was going to revolutionize the way that people do biology, and in particular our understanding of development. To go, different cells in our body do different things, and they do that because they express different gene products. And the ability to manipulate genes gives us tools to study that. What I decided to do was to study development in a developmental system where we had genetics, a very powerful tool to try and understand how it is that genes work, and that's in the fruit fly, Drosophila melanogaster. And I started working in Eric Weishaupt's lab at Princeton back in 1982 on a gene that we still work on in my lab, a gene called the runt gene, which gets its name because fruit fly embryos that are mutant for this gene die with very, very bad developmental defects. The body pattern is completely malformed. And I worked on that gene in Eric's lab for a few years, and then decided to take that gene with me, and my lab has been the runt lab in the universe ever since 1982, so that's quite a few years. What's shown here on the right, this is the kind of experiment we do in my lab all the time. These are actually pictures of fruit fly embryos. where we're looking at the pattern of expression of this gene, the runt gene, in an embryo that's 2 hours old, 3 hours old, and 6 hours old. The blue staining is cells that are expressing this gene. The white is where the gene's not being expressed. And the thing that grabbed my attention and the thing that's so wonderful about developmental biology is captured in this picture right here. This is a fruit fly embryo that's 3 hours old. At this stage, there's about 6,000 cells, and they're in a monolayer around this football-shaped embryo. This gene is being expressed in stripes. There's a stripe 4 cells wide here that's figured out to turn the gene on, and that's followed by a stripe of 4 cells that don't express the gene. 3 hours old, 6,000 nuclei, this little critter has figured out how to regulate gene expression in such a sophisticated pattern. So that's what's captured my attention, what I've been working on for the last score of years or so. Where's the stem cell stuff coming? When's he going to get to the stem cells? We're going to go there now. I worked on runts starting in 1982. It was of interest to people that were interested in fruit fly development, sex determination, body pattern formation, central nervous system development. There weren't a lot of other people in the world that were very interested. Maybe there was about a dozen of us. But it was the discovery in 1990 that there were runt genes in humans. That is where the interest broadened in our work. The first gene that was discovered was a gene on chromosome 21 in humans, so that's the Down syndrome chromosome. That's one of the most frequent targets for mutations associated with acute myeloid leukemia. That was published in a journal called the Proceedings of the National Academy of Sciences. The New York Times picked it up. They, on Fridays, frequently run sort of a little preview of what's coming out in the Science Journals next week. The caption of their thing was, Fruit Fly Leukemia Connection. My mother-in-law lives in Englewood, New Jersey. She saw this New York Times thing that mentioned my gene. She called me up and they said, I guess my daughter didn't make such a big mistake after all. So finally I was receptive into the family. So there was a connection to leukemia. Very quickly, over the 2 to 3 years after that, people went around and started isolating the runt-related genes in a lot of different species. What's shown here is this is a mouse embryo that's mid-gestation, maybe about day 14. And in this case, the blue stain indicates where this runt-related gene is being expressed. And where it's being expressed is the cells that are going to become the hematopoietic stem cells. So there, I said the word. There's a connection to hematopoietic stem cells. This is what got me in trouble. I said a graduate student in my lab, so I work on fruit flies, but one of my graduate students decided to go and look and see if there were runt genes in other vertebrate species. And what he looked at was Xenopus laevis. It's an amphibian, a frog that is a system where people have done developmental biology for over a century. And what Dan Tracy found was that this Xenopus, this frog runt gene, was also expressed where the hematopoietic stem cells were being born, as they were forming in the embryo. And so we published a paper writing this up, and one of the key words there underneath at the beginning of the article was this word stem cell. And so in 2001, when President Bush said, no more new embryonic stem cell lines, there was a lot of call for people to get educated about what was going on here. And in the Stony Brook University Hospital, the public relations person quickly scanned the database of scientists at Stony Brook to find out who knew anything about stem cells. And there was my article with the word stem cell as a key word. They called me up and said, okay, could you say something about stem cells? And so from that point, 2001 until now, the last 10 years, I've been asked to go out and talk to people about stem cells. I knew a little bit then. Over the last 10 years, I've sort of felt it was my mission to try and stay current with what's going on and be able to explain to the lay public what it is that the experts, the people that are doing real stem cell research, are working on. Before I leave runt, I'll just say that we know that there are 3 runt-related genes in humans. All 3 of them are mutated in different forms of disease, and in all 3 cases, there's problems in different lineages with cells deciding what to do, and frequently it's the stem cells that are doing wrong things. I haven't switched to working on human embryonic stem cells. I don't work on the mouse. I continue to use the fly, and I'll just make my sort of pitch here. The basic research, going and using model systems, so nobody's going to cure anything directly by studying fruit flies, but our understanding of how things works, we can learn a lot more in systems where we have more tools to play with. And I'll come back to that at the end. Basically, this pitch for basic research as being really, really important in terms of going to translational research and clinical applications for the things that we learn in these areas of biology. Okay, so a little bit of history on stem cell research. And so this is, I got called up, can you go give a talk to somebody? Back in 2001, I quickly started scanning, to try and find articles and things. And it was a time where things were, it was in the news quite a lot because of the Bush pronouncement. There was a very nice article in the New York Times sort of summarizing the history of stem cell research up to 2001. And I don't think you can read this here. I don't want to go through and force everybody to read all this, but I'll just point out a couple of the major points. Which one is that biologists have been playing with cells and trying to understand how cells come together to build multicellular organisms by taking things apart and trying to put them back together since after the Civil War. So since the mid-1800s, people had the idea that there were cells with the building blocks that make up multicellular structures, and that if we can take the cells out and do things, that we can learn something about how organisms are put together. And as far back as the mid-1800s, people were trying to recapitulate development in the test tube, starting off with in vitro fertilization. Let's take an egg and a sperm. We know that normally in the human body, those combine and you get a zygote that develops into an organism. What part of that can we recapitulate, can we reproduce in the test tube? And so people tried, starting in the mid-1800s. It took almost a century before there was the first success. So in 1958, I was 4 years old, I remember the day very well. It was the first successful in vitro fertilization with a rabbit. Another 10 to 20 years later is where it now first starts to work for people. The first human, Louise Brown, was born in England in 1978, the first in vitro baby. 1981 was the first success in the United States. I think now we all know that this is a pretty tried and true technology, reproductive technology, assisted fertilization, that there's lots of kids that are born that way. I have two nephews that were created by in vitro fertilization. It's something that, in the last 30 years has become quite commonplace. Okay, so I want to put that, so U.S. baby born 1981, contrast with that with another very, very important thing that happened with respect to stem cell research in the early 1980s. And this is now in the mouse model system. So not with humans, but in the mouse. Biologists going and studying early development in the mouse, and we'll talk about early mammalian development in a little bit more detail in a couple of minutes, but going to an early stage of development and showing that they could take some cells out of this young mouse embryo and that they could culture those indefinitely in a tissue culture dish. They'd stay alive, they'd continue to divide, and they'd divide and divide and divide and divide and divide, essentially forever. But then, at a later stage, you could change the cocktail of growth factors and drugs that they were exposed to, and you could get those cells to differentiate into different cell types. That was the discovery of these cells that are called embryonic stem cells. That in an early mouse embryo, you could take these cells out that were stem cells that would grow basically forever, that had the ability to give rise to many, many different tissues. So that's in 1981. What happened in the ensuing 20 years, from 1981 to, we'll go to 1998, is in the people that work on the mouse, this revolutionized mouse developmental biology and genetics. The ability to now watch cells differentiate before your eyes in a test tube, and to study cell division, and to engineer them, to take these cells and put DNA back into them to change their genotype, and then put those cells back into a mouse and get a mouse whose genotype has changed. That really revolutionized mouse developmental biology and genetics, including a spectacular study published in 1996 where people took some embryonic stem cells out and showed that they could differentiate beating cardiac tissue, that you could grow a heart in a test tube. Okay, so that's, in terms of regenerative medicine, that's pretty spectacular. That was 1996. We don't have that yet. So it's 15 years later, 14 years later, we're still not doing that as a therapy. The thing that I want to call your attention to here, 1981, we discovered there were these really cool cells in mouse embryos. 17 years later is when people showed that there were the same kind of cells existed in human embryos. So it's not that the mad scientists went running to do experiments the next day after these cells were first described. It was about 17 years that people in the mouse system realized more and more and more what the potential was of these cells and what you could potentially do. So then it was 1998 that James Thompson at the University of Wisconsin said, well, it's just what they're doing in the mouse system is just so unbelievable. I wonder if humans have the same kind of cells. And so what he did is he went to one of the clinics where they were doing in vitro fertilization. Typically when they do that, back in the late 1990s, you would fertilize a batch of maybe 20 to 30 eggs, and then you would pick out a few and implant them into the mother to try and get children, and then there would be leftovers. And so he got some parents to sign consent forms and said, okay, once we've done our assisted fertilization, we've got our children, we wanted to have 3, we've got all 3 of our kids, the ones that you fertilize and we don't need anymore, we're going to donate those to science, and you can use those to go and ask the human embryos have the same kind of cells as in the mouse system. So that was 1998. Because of the 17 years of work in the mouse system, it was pretty quick. He was able to show that they were there and that they had a lot of the same properties pretty quickly, in just a couple of years. And there was a rush. A lot of people started to say, okay, humans have these same cells. The potential is there to do a lot. A lot of people started doing a lot of things. And it really was going very, very fast. And that's when President Bush in 2001 just said, whoa, wait a minute. You know, where these cells are coming from is embryos that potentially have the ability to give rise to a human life that you're destroying to make these embryonic stem cells. We've got 80-some lines now. That's enough for the moment. Let's see what we can figure out with those 80 before we make any more. And so he had this edict to ban the use of federal money to generate any more human embryonic stem cell lines. At that point in time, in August of 2001, the first radio show I was on to talk about this, I actually agreed with him. He said, things are going really, really fast. We could use a cooling off period here of 6 months, a year, maybe a couple years to see what we've got our hands on and what's possible to do here. The thing that you know, I don't want to get too political here or too philosophical, but it was sitting in that same position for 8 years. Without, you know, he said, we're going to create a council, we'll get some people together that are really smart to talk about this and think about what we should do. Well, that council was never created. Nobody ever talked about what we should do. We basically sat in the same place for a long time. So that's up to 2001. Okay, so that's the history. We'll come back to the last 10 years and some of the recent advances in a few minutes. Before I do that, I want to talk a little bit about what the potential is here and get into some of the biology of actually talking about what a stem cell is, what do we mean by that. So the promise of stem cell research here, and this is a slide that's lifted from the director of the NIH in 2003, is that these cells, these embryonic stem cells, these cells that can grow forever and give rise to many, many different body parts, have the potential to treat lots of different human conditions. This is one of the things that I'd say people are looking to do is to treat these diseases. But these cells also provide the ability to study development in vitro, so as you watch these cells develop into different tissues, to see how that works. And these cells, since they're human cells, they also are the best place to go and ask about drug toxicity and drug specificity. We know that there are differences between human and mice. The FDA is not going to approve a drug based on a mouse passing clinical trials. It has to go through humans before it's given to the public. So there's a lot of promise there, and if we look at the numbers, the kinds of things that people think potentially could be treated with stem cell therapies is staggering. If you add this up, it's probably about 1 in 3 Americans that potentially have the ability to be treated for some condition. Heart disease is the #1 killer, and that's something that we think can be treated. I think it's mentioned in the press release that right now there's a clinical trial going on in Stony Brook where they're injecting stem cells into somebody that just had a heart attack, hoping that the stem cells migrate to the wound and are able to repair the heart. And so that's something that there's clinical trials ongoing now. Over the years that I've done this, I'd say there's 3 diseases that sort of stand out where the people that are the strongest proponents of stem cell research, and in particular thinking about the ethics of doing embryonic stem cell research and why it potentially is justified. And that would be diabetes, Alzheimer's, and Parkinson's, are the 3 that sort of stand out. Diabetes, we known for a long time exactly what the problem is. It's the beta islet cells in the pancreas are dying. You can't metabolize insulin properly. If you could replace that cell type, then you could cure diabetes. And actually one of the leading researchers in stem cell science in America is a researcher at Harvard who has got a lot of money from the Juvenile Diabetes Foundation to study exactly that process. And his interest in doing that, he used to also work on amphibian embryos, is he's got a kid with diabetes. And once he saw the potential, he said, okay, my lab is not going to change what it's working on, and we're going to try to cure diabetes. The other 2, Alzheimer's and Parkinson's, both of those diseases we know are due to neurodegeneration. Cells in your nervous system that are dying, that the nervous system starts to become dysfunctional. In one case, you forget where your car is or how you got here, which hopefully I don't get Alzheimer's during this talk today. Parkinson's, you lose motor control and you start to shake. Both of those tend to be late onset diseases, but if someone in your family gets that, it's very debilitating, not to just to that person, but to the whole family structure in terms of the impact on that person. I could mention that one of the leading proponents of embryonic stem cell research was Nancy Reagan, after Ronald came down with a neurodegenerative disease. And so she sort of changed colors a little bit there based on her own personal condition. And for all 3 of these diseases, now there's good reasons to think that yes, we are going to be able to treat these with stem cell research. People have made mouse models of all 3 diseases, And using stem cell-based therapies, you can start to ameliorate or cure the condition. And there's some proof of principle experiments with human cells showing that we're not that far away from potentially being able to do that. Okay, so that's why everybody cares about this so much, is that the impact on human biology and human medicine is quite significant. So let's step back and just ask, what is a stem cell? So a cell, for those of us that are multicellular organisms, this is one of the building blocks. It's one of the fundamental building blocks of our body. The other word here is the word stem, and when I went to give this talk the first time, I said, okay, let me go look up stem in the context of cell and see what it says. Wikipedia didn't exist then, so it wasn't possible for me to go there and look it up. But there's two sorts of definitions you see in Webster's. One is the main upward growing axis of a plant, so a long stem rose is a really good thing to be carrying around today. And the other is the main line of descent of a family, and I think this is really where the word stem cell comes from for the biologist. So what a stem cell means to a biologist is depicted on the next slide. This is the basic definition. In my undergraduate course where I teach this, this is the one definition. If they can't write this down at the end of the semester, then they're not going to pass my course. There's the basic definition of a stem cell. It's a cell that's going to divide and produce two daughter cells. So our cells divide by fission. One cell produces two, where at least one of the two daughter cells retains exactly the same properties as the mother cell, with the key property being the ability to divide and produce two daughter cells. So that's the definition of a stem cell. The ones that are in the news are the embryonic stem cells. And the early embryonic stem cells, the one green cell divides to give rise to two green cells. And so you get this expansion of the stem cell population. But at later stages of development, we have stem cells maybe in the nervous system or in the blood system, the hematopoietic system, where a cell will divide. It's a stem cell. It produces one daughter stem cell, but then it produces another cell that has a different phenotype that will start to differentiate and produce different types of structures. This is clearly, for multicellular organisms like ourselves, the ability to proliferate like this, to retain cells, they have the ability to divide and produce more cells is a key developmental strategy. So I want to talk about, we'll get back to embryonic stem cells in a minute, a couple of common examples of other stem cells that are in our body that we've known about for a long time. One of them, and in this, the juxtaposition of these two, I'll also sort of point out what are some of the key issues that stem cell researchers are facing in terms of trying to harness the power of these cells. So we all know about hematopoietic stem cells. These are cells that normally reside in your bone marrow, and they have the capacity to generate all the different cell types. If somebody has leukemia right now, one of the standard ways to treat it is you basically kill all of their leukemic hematopoietic cells. You give them a bone marrow transplant with a donor that's sufficiently well matched, and you recreate their immune system and their hematopoietic system using the stem cells that were in the donor's bone marrow. So hematopoietic stem cells, we know quite a lot about them. We have molecular markers that you could go to the bone marrow and you can actually purify cells and say, here's a population of hematopoietic stem cells that is pure, that I know exactly what genes they're expressing and what they can do and what they can't do. One of the big problems with hematopoietic stem cells, even though we know exactly who they are, we can't grow them in vitro. You think about it, if we could take hematopoietic stem cells out of somebody, out of their bone marrow, and put them in a test tube, and could recreate the environment of the bone marrow, you wouldn't need blood draws anymore. You wouldn't, like on NPR last week, they've been advertising that the snow sort of knocked out blood draw collections for about a week and we're falling behind. That wouldn't be an issue. If you could grow hematopoietic stem cells out of bone marrow, then you wouldn't need to do blood draws anymore. You'd create a bank of the different sort of diversity of hematopoietic stem cells you need to potentially give people blood transfusions. And whenever you needed some, you just take some out of the freezer and grow them up. That we can't do. And so that's a big issue. And that's in terms of developmental biology, a lot of what's being done with different types of hematopoietic stem cells is try to understand the environment that they normally grow in so that we can recapitulate that, we can reproduce that in vitro. Okay, so this is just a cartoon of hematopoietic stem cell. This is a self-renewing cell that gives rise to cells that are slightly different. These also can be stem cells. We can have myeloid or lymphoid stem cells that will continue to produce daughter cells that retain that sort of intermediate differentiation. But at the end of the day, in the hematopoietic system, we produce lots and lots of different cell types. Okay, so let me talk about a second example, and we'll see here a difference in terms of what we know and what we can do. It's the stem cells in your skin, dermal stem cells. So we know we have them there because if you cut yourself, or let's say you skateboarding and you go sliding across the pavement and you mess up a bunch of your skin, your body will fix itself. that the stem cells there will get called to arms, will produce more cells, and those will differentiate and replace your skin. The skin that's on your surface now, those cells are already all dead, and they're being replaced constantly from the stem cells that are underneath. What we know in this case is that you can go to somebody's leg and you can do a biopsy, take out a little plug, the dermis, about the size of a dime. And in this case, we know the culture conditions for growing those cells. You can take that out, put it in a culture dish, and in 2 weeks you can grow enough skin in vitro to cover the entire human body. So this little plug can produce enough cells that are doing the right thing. And that's the person who I learned this from, is the director of the Living Skin Bank at Stony Brook. The major job of this skin bank, when a burn victim comes into the hospital, is that the doctors in the emergency room need to keep that guy alive for 2 weeks until they can grow the skin to give the guy the skin transplants and put it back on. And so in this case, we know how to culture the cells. We can grow them, but in terms of the stem cell biology and understanding who's doing what, within this plug of tissue that you're taking out, there's still controversy as to which is the stem cell. So we don't understand the biology well enough to really know which is the player that's doing this. So we know now in adults that there are stem cells in basically all the major body organs. And if we think about it for a minute, that probably has to be true. You live now for maybe 80, 90, we're approaching where more and more people are living to 100, that cells in all of your tissues are dying off with time and they need to be replaced. And if they're not replaced, then you're going to lose that organ. And what's happened over the last 20 years is people going to different organs and finding that there were different types of stem cells. Back in 2001, everybody thought, well, the one place we know there's no stem cells is the nervous system. Because if you break your spinal cord, it's not going to be repaired. Those cells don't exist there. Well, we know now that there are neural stem cells. And actually, there are neural stem cells in the peripheral nervous system as well as in the brain. And these cells can give rise to lots of things. And the trick now, with spinal cord regeneration, there's clinical trials ongoing where they think it's actually possible to do this. And the key is that you need to be able to get the cells there quick enough.